Thank you, Mark. Thanks, Anne, for the invite to give the Sir Julian Huxley lecture today. And when Anne asked me, said we would like to invite you in the summer because you're not only describing species or using described species, but you also apply them for further research. This shows how you can use and how important species descriptions today are. And I hope that my lecture that I then put together and realized it is not that easy to do is actually answering some of the questions, and I can actually do what I said I would. So the lecture today is about the marine biodiversity of the Southern Ocean. The Southern Ocean is the area around the Antarctic, and in modern times we know it's cold, but it hasn't been like that. Like these images from the past, from the Cretaceous, shown that the Antarctic around 150 million years ago was quite warm, and the seas were quite full of large animals like the mesosaurs and huge cephalopods. Nowadays, and since 150 million years ago, here we see the comparison between, on the left-hand side, a Cretaceous fauna in the benthos on the seafloor, and on the right-hand side, the nowadays modern Antarctic shelf fauna. Antarctica in the past and today, so 125 million years ago, the Antarctic was still a part of Gondwana and connected to the southerly continents and still had the warm climate, while nowadays it's centered around the South Pole and has quite different environmental and abiotic factors. So the Antarctic today, in the summer, on the left-hand side, is, when it's seen from space, is very white, very cold. Most of the land is covered by ice. And in wintertime, it gets even whiter. Here, the sea is not open anymore, it's covered by sea ice. And this is to remember, seawater is only freezing at minus 1.9 degrees. This means that all this area around the Antarctic is extremely cold in the winter months. What else is influencing the Antarctic? The Antarctic is influenced by extreme winds. We have the westerly winds that go all around the Antarctic, and they actually not only drive the winds on the surface, on the land, they also drive the ocean circulation because they take the ocean surface and with them the underlying layers with them. So in the Antarctic and around the Antarctic, we have a very strong circular current system, the Antarctic circumpolar circulation, which can be seen even from space by different also colors in the temperature when you have satellites using sea surface temperatures, for example. And this is seen in the next images. On the left-hand side, we have the sea surface temperature, and on the right-hand side, the seafloor temperatures. If I first describe the surface seafloor temperature, we have the tropical zones where we start at degrees of about 20 degrees, and then we come to the subtropical front where the warm waters and the cold waters start mixing, and we go to around 4 or 5 degrees until we come to the polar front where we have the very, very clear divide between warmer waters and the 0 degrees. And it's almost, you come through that, and when you cross the polar front, those few kilometers of sea line, when you're at sea, you feel it. Within 10 minutes, you can go outside without a jacket to the moment when you go outside, and you definitely need your down jacket and a woolly hat. So closer to the continent, we have temperatures all year round of about 0 to minus 2 degrees. And in some areas of the Antarctic and in the Antarctic deep water, we have extremely cold waters. You can find waters in the Antarctic, in the Ross Sea and in the Weddell Sea that are down to minus 4 degrees. In general, you expect those waters to be frozen, but because of the high salinity of these waters and the pressure on them, it is still liquid. But if, for example, you have a little sand grain or something that goes around across the water item, they can suddenly form ice cubes or ice plates on the seafloor. So sometimes in the Ross Sea, you can see images of really, really nice platelet ice that suddenly forms on the bottom of the sea. But what do we do about the Antarctic marine biodiversity, because that's the actual subject of the talk and not the environmental conditions. We have a register of Antarctic marine species that is run by colleagues in Australia and has a large community of global taxonomists being in charge of their relevant groups to verify the Antarctic species we know in the marine system. So to date, we know that there are over 9,000 species living in the Antarctic waters. If you get confused because I say we have 8,236 taxonomically accepted species, this means that these species have been checked by our taxonomic experts and all are accepted species in the Register of Marine Species. Well, the extra 2,000 species for the listed species are species that are looked at by taxonomists that, for example, said CF like somebody else. So they are different, but they haven't been confirmed to be an accepted species yet. And then we have another 9,000 registered taxa. This means they are not taxa in that it's a higher qualification. It's something that we would call morphospecies, where we haven't done the descriptions yet. So it shows how large the number of known species, even if undescribed species, are in the Southern Ocean. And how are they distributed in the Southern Ocean? Because if you see any documentaries around the Southern Ocean, it is dominated by penguins, seals, and whales, sometimes even seabirds. And of these 9,000 described species, they only take 90 species. So it's a really, really low number of them. And this is where I advocate my group of the species that I'm interested in. If we go to the pelagic area of the sea, this is everything from that lives in the water column, from the surface to the deep sea floor. But above the sea floor, we have around 700 species. They are often distributed all around the Antarctic and flowing along with the water masses. And now we come to my favorites, the benthic species, of which by date we already have over about 8,000 species that we know about, plus most of those undescribed 9,000 species actually live in the benthos. And it's incredibly rich. So here, and I hope it plays, is a video showing you an area in the Weddell Sea with the different species on the seafloor in 300 to 600 meter water depths. So on the UK shores, this would already be counted as deep sea because here our continental shelf break is at 200 meters. In the Antarctic, it can be as deep as 800 to 1,000 meters. And you can see it's quite a dense populated shelf with various interactions, like you can see here, between an octopus and an icefish. Or we have huge glass sponge communities that are used as secondary habitats, and they are biogenic habitats. Here you see crinoids, the comatolites escaping from the video from the ROV. They quite often show this swimming behavior when we come along with our nets and our cameras. Here you have a very nice community, dense community of different types of sponges and hydrocorals. So what else is special about the Antarctic? I said we have such a high benthic and pelagic biodiversity. Well, because the Antarctic had been separated from the rest of the world by the circumpolar current, we had quite a high number of endemic species. So the endemism rates in the Antarctic, this means species that only occur south of the polar front, are ranging between 23 and 100%. And this 23% in the Tanaidasians is quite unusual. We often have endemism percentages between 60 and 80%, as you can see in this row. So it shows how special the Antarctic fauna is, and that if the Antarctic would disappear, we would lose all these unique species. The pelagic species in the Antarctic generally show a wider regional distribution. They often occur all around the Antarctic based on the water mass. While the benthic species, and especially the benthic brooding species, show very restricted distributions. And this is also shown, can be seen for example, in the isopods, where we have over 900 species in the Antarctic, and sometimes they are only found in one particular site of a few kilometers. And if you take a site again in the same depth, 10 kilometers away, you find a completely new composition of species within the same genus. Over the last 20 years, we had quite a shift in paradigms that we had. When I started studying the Antarctic, we learned that the Antarctic circumpolar current, which started with the separation of South America and Australia from the Antarctic, was a barrier for dispersal and gene flow, and we since then learned it is permeable. You are able, as a marine animal, to cross it at different time scales. We sometimes still have crossings today, especially with animals and taxa that are linked and living on kelp or floating objects that through currents and eddies can pass through the polar front. We had the paradigm that the Southern Ocean marine fauna has a history of in situ evolution, that it all came out from itself, that it started after the last glacial maximum. And with studies in 2009 by Conway et al., we could show that the taxa in the Antarctic have a very long persistence. They stayed through the last glacial maxima. They didn't get extinct. But we also had a very rapid quaternary radiation in many groups of the Antarctic. And something else I grew up with in my science career was that decapod crabs in the Antarctic got extinct in the Eocene. And this was the reason why the modern Antarctic benthic fauna is quite archaic, that we have a lot of these fragile taxa, like the stalked crinoids and the comatolid crinoids that you can find in large abundances, because they don't have these shell-cracking predators like the crabs. Recent studies by Rowan Whittle actually showed that until the Miocene, we had quite significant decapod components in the Antarctic benthic communities. So these archaic states that we find nowadays is actually not just because of the disappearance of the decapod crabs. Coming to the points I showed, I wanted to give you some examples on the gene flow across the Antarctic circumpolar current. My example is Parabolasia corrugatus, a very pretty nemertean that can grow up to 0.5 meter length, 3 centimeter in size, and feeds on anything that it finds. When I was an PhD student on the Antarctic base, one of our monthly tasks was to collect 20 of these specimens together with 20 brittle stars. You went down diving, picked them up, put them in your one collection bag, came back up, and you realized you lost all your brittle stars. But in your nemertean, you could see that they had eaten them, and you could actually count the discs. So, coming back to the science, a group of colleagues around Thornhill collected Parabolasia in South America, so around the tip in the Magellanic region on Birdwood Bank, the South Georgia, South Sandwich Islands, Bouvet Island, and the Antarctic Peninsula. They collected 86 specimens and did analysis of the CO1 and 16S gene and dated them, and realized while they found 2 clades of Southern American and Antarctic subantarctic island populations, they realized that this clade separation only occurred about 4 to 14.5 million years ago. The circumantarctic current established about 28 million years ago. So since the establishment of the circumantarctic current, we had different taxa crossing it at different times, coming in and out of the Antarctic. And this pattern we have seen over the last 10 years in several taxa. We also had the long-term persistence in the Antarctic. The Nymphon australia is a sea spider, and we have records of sea spiders for millions of years in the Antarctic. We had to look into this particular small, abundant species called Nymphon australia. In the initial study, my colleague Claudia Arango had 67 specimens that she looked at for the barcoding gene on 16S and CO1. and realized Nympha nostrale is actually a real circumantarctic species, despite being a brooding species. So how do they get around? How did they do that? And was it just those 67 specimens she had? So through Anna Soler-Membrevis, we were able to extend the data set. We used 364 specimens, extended our genetic locus set, and did actually a demographic history. And what we also had this time was getting animals from all around the Antarctic. And we realized it didn't matter where they came from the Antarctic. They have a very, very close haplotype network. All of them, doesn't matter if they are from the Ross Sea, the opposing Weddell Sea, from Bouvet Island or other subantarctic islands, they all grouped together. They had a high number of unique haplotypes, but we had some dominant types. And we looked at how did they survive in the Antarctic? How did they survive the last glacial maximum? Because it's a species that prefers to live on the shelf, in the shallower waters. The highest abundances are found between 400 and 800 meters. So with our demographic history analysis, we realized we had 3 groups that were really centers of radiation, which were in the Antarctic Peninsula, in the East Antarctic, and then in the Weddell Sea. We realized that the high diversity in the Antarctic Peninsula and the East Antarctic related to multiple contraction and expansion events associated with deep sea refugia, which happened about 21,000 years ago, so during the last glacial maximum. Saying they had deep sea refugia means during the last glacial maximum, when the ice went all across the Antarctic shelf down to the shelf edge, the species was pushed down into the deep sea as far as it could go, and then afterwards, when the shelf ice retreated, came back up onto the shelf, and then continued. The diversity in the Weddell Sea reflects a more recent colonization from one shelf refuge somewhere in the Weddell Sea, and we are now trying to find out where do these shelf refuges exist, because this means that when we have the last glacial maximum and the ice shelf continued to the continental shelf edge, somewhere underneath the ice shelf, we had pockets of seawater where animals were able to survive. We have our so-called archaic Antarctic benthic state with the high abundances of brittle species like the stalked and the unstalked crinoids. So Linnaeus had a look at the phylogeny of the crinoids in 2013, but she only included, I say only because it's a huge data set, metacrinoids and comatolids. The oldest known crinoids, the isocrinoids, were not included. The stalked crinoids, like the isocrinoids, are known since the Ordovician, so for more than 400 million years. We know we had a very high number of stalked fossil crinoid diversity around 200 million years ago, and today stalked forms are normally only found in the deep sea, except in the Antarctic, where we can find stalked crinoids in shallow waters as shallow as 50 meters. We find them in the deep sea as well, and we do find both stalked and unstalked forms in the Southern Ocean. So, a recent study by Whittle showed, when she looked at the fossil history of stalked crinoids in the Antarctic from the Paleocene to the modern, in the Antarctic and in Australia, and they were looking at microfossils. So they were not looking at the large fossils that you can see with bare eyes. They were actually going through the sand that was sitting in museum collections to find the little stalk of the crinoid stalks to identify those. And they realized they found 30 new isocrinoids, these really old-fashioned fossil species, until the Eocene and Oligocene in the shallow waters of the Antarctic and Australia. But these isocrinoids decreased in abundance in the Eocene at the same time when the comatolids, the unstalked crinoids, appeared in the Antarctic. In parallel to this study, now the first living isocrinoids, this really old group, has been found in the deep sea of Japan. And this for the first time will enable crinoid specialists to look at a molecular history for the Southern Ocean crinoids, because we now can look at the old-fashioned fossil crinoids, and we have a very good constrained fossil background of the crinoid Southern Ocean species. Over the last 20 years, we also found new habitats in the Antarctic that we didn't know about. We found hydrothermal vents with low venting system and high venting systems. We had discovered methane seeps under ex-ice-covered ice shelf habitats as well as in the open sea. And we discovered under ice habitats as such that animals can live like they did in the last glacial maximum in the refugia time. They live under the ice shelves. So on the hydrothermal vents, some of you might be familiar with the black smokers, which are the high temperature venting. And we also found some low temperature venting. I will start to explain the low temperature venting, where you can see shimmering water coming out. You might see small chimneys coming up from the seafloor, quite maximum of 1 meter in height. And when you look at around the fauna, you don't see any vent endemic genera visible. You have the normal Southern Ocean benthic elements. You have no dense populations. You do find some siboglinid polychaetes, which do like the high organic content in the area, the high sulfides. We have a huge amount, or larger amounts, of ophiurids, but brittle stars are very common on the Antarctic seafloor in general, some anthozoan and some sponges. So they look like a normal Antarctic soft bottom seafloor to me. The high temperature venting and black and white smoking areas are completely different. We measure temperature of up to 380 degrees in situ on the hydrothermal vent sites. And we find the typical vent communities with vent endemic genera, which are visible in the Antarctic through the Hoff crab, the Kiva crab, the Yeti crab, little limpets called volcanolepers, the seven-armed starfish, Polisterias, and what we've realized, all of our Antarctic species on the vent sites were new to science as well. And the typical Antarctic Southern Ocean benthic fauna comes in to the diffuse flow areas of the vent sites, being attracted by the high abundance of food. And we have very dense populations. And in the next slide, I want to bring to your attention the characteristics of the East Scotia ridge vents, where we found the high abundance of more than 1,000 individuals per square meter. We have high amounts of endemic species, and in general, the species diversity on the vent sites directly is quite low, with 11 to 13 species, while in the diffuse flow, where the normal fauna comes in, we could find between 19 and 35 different species. Quite interesting for us was to realize we don't have any tube worms, any large tube worms in the Antarctic vents, none of the albinocarid shrimps that we know from the North Atlantic vents, and none of the Batomodilus bivalves. What we have instead is the large Yeti crabs, where the large males can grow up to 12 centimeters. We have huge amount of stalk barnacles, where the stalk can grow up to 1 meter, half a meter to 1 meter in length, while the patellum, the head of this, is only 1.5 centimeters in size. We have large peltospirid gastropods, of which I will explain in more detail the fascinating ecology. Large sea anemones, and that are one of our larger predators in the area. 7-armed predatory starfish that feed on the Yeti crabs, for example. Our hydrothermal vents, where you can see one on the left-hand side here, show a clear zonation of the different taxa you can find on it. So we were wondering what the reason for this faunal zonation was, because everything in here in the image, which is taken here, is an assemblage characterized and dominated by the large barnacles. Well, the singleton large male Yeti crabs were only found really close to the black smokers. So we had to look what made the pattern, and we realized the distance from the temperature, from the hot venting, is actually making our microzonations. The closer you are to the black smoke coming out, there we only find the really large males. The further you go away, the further the temperature and the biogeochemical composition is changing. We have different communities dominated by male and female kivas going to the smaller specimens, going to an area dominated by our peltospirid gastropod, coming to the diffuse flow area with the barnacles, and then into the off-venting side with the large anemones and the peripheral fauna. On the vent sides, the most charismatic species that we found, and the one that got most of the media attention, was the Yeti crab, which here on the bottom you see one of the large males compared to a smaller female. And as I said, they can grow up to 10, 12 centimeters. They are characterized by very dense bristles on their underside and on their arms. And these bristles, when you look at them under specialized microscopes, you see that they are all covered by bacteria. So what the Yeti crab actually does, it harvests, has its own allotment, its own food farm on its underside. And to feed, you can see them using their claws going along to their bellies and feeding on the bacteria that are growing on them. And we had a look, because this is known from other vent crabs like the Kiva hirsuta and the Shinkaya. And our microbiologist, Katrin Linse, had a look at the composition of the bacteria. And she realized that over 90% of our bacteria are forming a single clade that is only existing in the Kiva tyleri, in the Antarctic one, while we have some other strains that are linking with the clades that are found in other vent crustacean bacteria that are living on the bristles and that they feed on. We had a look at the ecology of the Kiva crab, because we realized the big males, where we would estimate the alpha males that make the population, are only sitting on the top of the hydrothermal chimneys, where you actually don't have females. So our first idea was that these are the alpha males that maybe are the dads of most of the offspring, but we have never seen them moving. When we came back 3 years later, we interestingly had the same number of large males sitting at the same position at the vent sites. We can't say if it's the same animal, but it's interesting we still didn't see any really large ones further down. We then, going a bit further down from the really hot vents to an area of 25 to 40 degrees, where we have a mixture of males and females. We have these large communities where they sit all on top of each other. And interestingly, we had a lot of females that had just molted in the area. So we're assuming this is where they are meeting up. And further away, we realized in the periphery, we had some female crabs that looked quite dirty and yellow, and they were actually just before molting. And we also had some females there that were egg carrying, and so when they don't like the hot water when they are carrying their eggs, because the really hot water could damage their eggs. When they go out, when they are molting, just after the molting, which means they are able to, for the next mating, they come back, meet up with the males, and then for the next time stay in the slightly warmer waters that don't harm the offspring. To have a look at how one of those Yeti crab looks, I have a slight problem with the video, so I need to go out, but I wanted to show you how one of these Yeti crabs in the warm 24 to 40 degree water looks. You see the large male here, and on this one it's living in a very fruitful, well, bacterial environment, because it doesn't only have the bacteria growing on its underside, it also has very, very long bacteria growing everywhere on it. So let's decide where we don't see much of the movement of the specimens, and when I'm going back next April, I'm interested to see if we see the same type or size collection of the specimens still at the same sites. The next new species and genus that we found was the large Paltus spiroid, which I wanted to bring in to this talk because this genus and the species was described in the Zoological Journal of the Linnean Society. We have the species lives in huge groups. On this image, you would have about 3,000 species. They are being stacked on each other. We wanted to collect 50 with a suction sampler. And we thought we were quite good by counting each of these large specimens. They're about 3 centimeters in diameter. When we counted them afterwards, we realized we had 150 because between the big ones, we had really tiny ones down to 2 millimeters. By looking at the species, our Gigantopelta and its sister species from the southwest Indian ridgevents look quite similar to the scaly foot Gastropod, which is quite famous in the malacology world because it's able to form some iron sulfide scales on its foot. So we looked at this, and the first indication was at sea, oh, are they in the same genus? But work by Chongqing realized they are not in the same genus. They are not even very close to each other in the tree. But it's a quite interesting species where we have work ongoing. So Chongqing continued with working on the Gigantopelta chessoa, and here did a 3 tomographic anatomy of the species where you very nicely can see the nervous and sensory system, the in red we should, then the digestive system, and very interesting here is the esophagus and the esophagus gland. And in this specimen, it is extremely small and in the size of a normal esophagus gland if you're a malacologist and know how to dissect one of these snails. So for me, I now know that this specimen is a very small juvenile. Because when Chong had a look through size cuts from them, from specimens of 2.2 millimeter to 2.3 to 23 millimeter in size, he realized that their inner anatomy is completely changing. And what we have as the esophagus gland at the really small animals and is extending with their growth to a new organ that he named trophosome, because it's comparable to the trophosome in the large zipglinid worms. It's full of bacteria, and the animals, when they are larger than around 7.5 or 7-8 millimeters in size, only live off the bacteria in their glands. And this is shown in this graph where we looked at the complete digestive system and then the amount that this trophosome organ is making of the system. And so these species, well, when they start up as youngsters, they graze, but at a certain time, the full ecology is changing. They go through a food metamorphosis from grazing on bacterial mats. They change and implement bacteria somehow, and this is still an open question on how they do it, into this former esophagus gland, which now goes into this trophosome. And again, I wanted to show you these. I say in action. Here, you see in the video around this area the slightly shimmering water. So again, they are in waters that are warmer than the ambient temperatures. They don't move much. If you look at the animals that you see out here, they just seem to sit. If you sit there for an hour, you might see a movement of the tentacle, but not much. Oh, that wasn't expected. Sorry. So the next habitat we discovered were the methane seeps. We had methane seeps under the Larsen B ice shelf. The Larsen B ice shelf collapsed in 2002, and we only had a few meters of water between the seafloor and the bottom of the ice shelf. So in an area where we have extreme warm sediments and granites in the seafloor, there's more warmth and hydrofluids coming up, which enable the methane then to come through into the sea, and because they can't be driven away, the methane, with the open currents, the concentration comes up, and we actually were able there to find assemblage of seep and vent communities, humusynthetic communities of large vesicomyid clams. We have dense abundances in small patches of them, but about 2 years after the ice broke out, we couldn't find any living clams anymore. Then, on the other hand, we discovered methane seepage around South Georgia. We discovered more than 1,500 methane seepage sites on the South Georgia floor and a cruise last year. And you can see one of them here with the bacterial mats. We don't have any vent endemic fauna. Even the normal Southern Ocean benthic fauna is almost absent from the community. We don't have a dense abundances. If we find some animals in the methane seep areas, it's siboglinid and dovelid polychaetes, some anthozoans, and the brittle stars that we have everywhere in the Antarctic. And this is the last of the videos. But what was very interesting when we were at the South Georgia shelf was the huge abundances of krill that was swimming around the methane outflow sites. And here we tried to take a sample throughout this methane seepage sites, throughout the bacterial mats that are coming up like mounds, and you will see just by the way the perspex tube is going in, you can almost imagine how the consistent of that sediment is. And in the background, you already see it is very black, and when the samples came back, it was so smelly of sulfites. So we had to really protect ourselves with respirators of working with the samples. But this is the interesting, we have methane seepage in 2 areas. One is able to host the humusynthetic communities, and the other one doesn't. The latest habitats are the under ice shelf habitats. The Antarctic is the only place in the world that has large ice shelves that going over the continental shelf, and the different ice shelves are marked on this map. In the recent years, and in 2007, for the first time, we had discoveries by drilling holes through the Amery ice shelf of discovering sea life under the ice shelf that is several hundred meters deep. So is this the place for the last glacial maximum refugia for the benthic fauna or not? And this question is now still being asked. What is the fauna that we find there? Recently, the ice shelves have been in the media because we had huge calving events. The entire Larsen A ice shelf here, which is not even marked, collapsed in 1995. The Wilkins ice shelf here collapsed and disappeared completely in 1998. Larsen B on here broke out in 2003, and Wilkins, a second part, the last part, disappeared 2008, and last year we saw the huge calving event of the Larsen C iceberg A68. And this is where we go, what is living underneath this ice shelf? Are they our old last glacial maximum refugia? Do we need to get in and collect and study the fauna that is in there? So in the study by Martin Riddle in 2007, when they went into the Amery ice shelf, they had a station 100 kilometers inland or on shelf from, this is 100 kilometers distance between the shelf ice break and the inner station. And underneath on the seafloor, they discovered a vivid community of suspensions feeders, of sponges, bryozoans, and hydrocorals. What I find interesting is they had only 480 meters of shelf ice thickness. So they had about 400 meters of water between the ice shelf and the seafloor. So actually water circulation was able to get in there. And this is why I think this suspension feeder community was able to be in the Amery ice shelf. I don't think we have that in all the other ice shelves. Because what we have, and that is a pre-collapse image of an under ice shelf habitat, is we often only have a few meters, maybe 10 meters, between the sill here and the ice shelf on top. So we don't have the currents coming in. So this is why we have this unique community here. And after the ice shelf collapsed, like we have seen it in various places in the Antarctic, the whole community that lived underneath it will be changing. But the changes are not only for the under ice shelf community, it's also for the deep sea community along the continental slope and in the Jess and deep sea basins, because the whole ecosystem is changing. And as I said, we had the Larsen, the Antarctic Science Research Community, and especially the Commission for the Protection of the Antarctic Marine Living Resources, realized that these Antarctic ice shelves are unique on the planet and that they are required to be studied and kept away for research from fishery impact. So if an ice shelf collapses, it is protected for 2 years for research, and then after biodiversity survey, it can be extended for another 10 years. Sorry for the mistakes, it's not 20 years, for 10 years. So last year, this 150 by 50 kilometer large iceberg A68 broke off. I've spoken a lot about it. It's 4 times the size of Greater London. And this one here is around 400 meters deep, but the waters around it is only around 430 to 450 meters deep. So this area underneath here didn't have much area to actually get any ocean currents in to establish a suspension feeding community. For the first time last September, CAMLA designated a special area for scientific study, which means this area is now protected for another 10 years from fisheries and only available for research. And international expeditions are on the way to study it. The British research organizations gave us funds last year to try to get into Larsen, but this very beautiful pack ice field with these nice press ice ridges that you can see was not as beautiful when you tried to get through it. They were 4 to 5 meters high in the sea ice, plus going up to 10 meters in the press ice ridges, and we had to turn around and come back. But this year, in the coming season, there are again 2 or 3 international expeditions trying to go into the Larsen Sea ice shelf to study the under ice areas. And in the last months, we had extreme westerly winds and warm westerly winds in the Antarctic winter that pushed all of this ice out, which is now on the way to South Georgia. So the expeditions going in January won't be held up by this ice again. And this brings me coming to the threats in the Antarctic. What are the threats for the Antarctic marine fauna? We have the climate change. The Southern Ocean is warming, the glaciers and the shelf ice are retreating. We have the fisheries as a threat. In 2004, Murray and Butterworth said, the Antarctic region is the region where the largest human-induced perturbation of the marine system in the world has taken place. In general, we think the Antarctic is this pristine continent and nobody has been there. But we only need to go back 100 years with all the whaling and all the sealing. Because through taking the whales and millions of whales out, the whole ecosystem has changed. And then we have the recent fisheries for krill and fish, which is CCAMLR regulated. And through that, we also plan with the fisheries to implement marine protected area. So with the climate change, the ocean is warming over the next 100 years. We all have heard this. How will this affect the Antarctic? Will it affect the Antarctic? The Antarctic is cold. And yes, it will. It might not warm up as much as the North Atlantic, but it will still warm up quite a lot, by almost a degree in the surface waters. And how, if you're an Antarctic benthic or an Arctic animal, what can you do? You either adapt. You adapt, but can you adapt in the fast time in just 100 years? Your other possibility is you go into the deeper waters, because your deeper waters should be colder than the surface waters. You could go to the poles. Or you'd go anywhere and go extinct. So to find out what we do with the climate change, if we, from the Antarctic benthic community, can say something about the effects of temperature change and seawater rise, temperature rise in the Antarctic, we looked at almost 1,000 benthic species that had more than 20 records in the Southern Ocean and adjacent areas, and are collected above 1,000-meter water depths, so that they are still being affected by the sea surface and the sea temperature rise, from 40 degrees south to the Antarctic continent. We looked at 10 phyla where we had the best taxonomic confirmation on the species and the knowledge on the species. We took the data from the SCAR Biogeographic Atlas of the Southern Ocean and realized only of these 1,000 species where we have a good record and a good ecological record, only 390 species occur only south of the polar front, 430 cross the polar front, and 150 live not only north of the current polar front. So we had a good range of species. We used different climate models, 19 different climate models, and the RCP 8.5 mean assembler for the highest seafloor temperatures in 2099. So we grouped our 1,000 species along the temperature ranges, and by the temperatures from -2 degrees to +19, which was the range we could find in our 1,000 species, and see what are the limits of these species, where do they occur, and when the temperature is changing, which species will be affected. And the blue line here shows you the current south of 60 and the south of the polar front. In 100 years' time, this temperature of about 2 degrees is shifting to 3, and the polar front from 2.5 to about 3.5 degrees. So that shift by 1 degree in temperature is changing the composition of the Southern Ocean marine species. Most of them have a decreasing area. So they will be pushed further to the poles, and the region they live in will be smaller. Only a few species that can live in warmer areas actually extend their species range, because they can live further north as well. That is one of our threats. The other threat for the Antarctic is the fisheries. And yes, we have the Convention of CCAMLR that aims to conserve marine life in the Southern Ocean, and however, it does not exclude harvesting carried out in a regulated manner. And the Antarctic marine living resources means any population of finfish, mollusks, crustaceans, and they also look at living birds. They don't look at seals, and they don't look at whales. The Commission have split the Southern Ocean in their different zones, so they can discuss the fisheries in various zones. And this Antarctic fisheries zone is the size of about Europe and North America. And it's about 10% of the world's ocean. And at the CAMLR meetings, you have about every country in the world trying to have their say and potentially get their take of Antarctic fish. The most important fishery is for Antarctic and Magellanic or subantarctic toothfish. The Dissostichus, which you can see here, it's a species that can grow over 2 meters in length, can weigh over 100 kilogram. Each kilogram can bring you up to $25, $30. So it's a highly valuable fish. It is very long-lived. You could live with more than 50 years. They only get mature with 15 years. So it's a very long-living, late reproducing species. And we have the fisheries for the subantarctic around South Georgia, the Falkland Islands, and all the subantarctic islands. And we have the Antarctic fisheries, especially concentrating in the Ross Sea and marginally in the other seas. You will notice that here, in this area of the Weddell Sea, there is currently no toothfish fisheries for commercial fishing. It's only there for research fishing. But there are several countries that want to go into the commercial fishing there. And this is where the marine protected areas come in. And for the Antarctic, CAMLR is the way into policy to bring the regulations into the fisheries. But to propose an MPA, and we have a couple of MPAs. There's a marine protected areas with very regulated fisheries around South Georgia and the South Sandwich Islands. But the reason for that is they have their own government and are outside of the CAMLR regulations. And given the Antarctic Treaty, the Antarctic is not state-owned. It's all, no country can claim a certain part. We have a small MPA around the island of the South Orkneys, which was declared and accepted because of the high amounts of seals, penguins, and birds feeding on krill. So the krill fisheries is regulated there. We don't have any regulations there for the benthic work. And in the last year, 2016, the Ross Sea MPA has been adopted, and adopted by all countries that are represented in CAMLR. But you will see, it's quite interesting that this area is not there. It is not protected, because this is the area where the fishery businesses are in. And when they tried to protect that area as well, they failed with getting the rest protected. So if you want to protect an MPA, you need to propose it to CAMLR. You need to produce a scientific report, including a report on the biodiversity. You need a spatial management report, research and monitoring plan, and then that proposed MPA needs to be adapted and agreed by all members. And in the recent years, the proposed MPAs for the Antarctic Peninsula, the Weddell Sea, the East Antarctic, all have not been agreed because there are 3 pushing countries in the world that are very interested in getting the fisheries in and harvesting the toothfish. And this is, I was involved in preparing the proposed Weddell Sea MPA, which is a German-led and EU proposal to CAMLR to show that all the European countries, including the UK, are behind it and are working on this for a decade now. We had to look at all sampling spots in the Antarctic, all biological data that we have. We had a look at the different zones that we find in the Antarctic, what bioregionalization we have, and on the right-hand side, you see the areas where we actually have spots of Antarctic data points for the benthic seafloor. Because you can't go in everywhere, and it's a huge area. And if you want to know about the biodiversity, you only have a spot, and then you estimate. And we can do now species distribution models. But those countries that don't want to agree the MPAs, they just say your data are poor, you don't know your species. And this is where I say this is why it's even nowadays so extremely important to have good species identifications for doing meaningful assessments of the Southern Ocean marine biodiversity because of the change in our climate and because of our conservation issues. Most of our Antarctic marine and pelagic benthic species will face distributional range changes in the coming decades. And the designation of the Southern Ocean marine protected areas, which we also require to protect those species, to not having them under two threats, the fisheries and the climate. We can't do anything about the climate as such. Yes, we can, but the temperatures will rise. But we can do something about the protection. And for this, we need to know what species we talk about. Because what we get at the moment back from the countries that don't want to agree the MPAs is we have our data poor and we don't know who we have. And this is why I still think that taxonomic and systematic research is so important. So I'd like to thank my colleagues and the funding agencies that enabled me to give the presentation today. I'll thank the Linnean Society and the Systematics Association. And I'm welcome to you for coming and asking questions.